invitation. I'm very happy to be here and I'm looking forward to the next 2 days and of course to collaborate with you. But for now, I'm happy to share my presentation with the title Introducing Laboratory Automation, Benefits, Problems, and a Possible Answer for Everyone. And as Domini said in advance to me that we are today a very heterogeneous audience regarding the level of knowledge about laboratory automation, I hope that I prepared my presentation in a way that it will be interesting for every one of you. So first of all, I want to show you a graph about the life expectancy in Estonia and the development of the life expectancy in Estonia from 1950 to 2022 with a forecast to 2050. And I think it's very obvious that the life expectancy increases and people get older and older. And this fact applies to the whole Europe and also the entire Western world. Reasons for this development are hygiene aspects, education, improved working and living conditions, but a big part is the medical progress. So on the one hand, in improved therapy methods or possibilities, and on the other hand, we have a better prevention of age-related diseases like cardiovascular diseases or cancer. And the reasons for that is the development of drugs that improved, as well as the prevention by early diagnosis. So the development of drugs, nowadays we can bring faster and more efficient drugs on the market that are safer, and we have the prevention by better, faster, and possible frequently diagnostic nowadays. And this is related to laboratory automation, which is the integration of automated systems in the lab to automate manual processes. And as Michael is an expert in drug discovery, I will focus in my presentation on the diagnostic nowadays. And therefore, I want to show you some facts about the daily routine of a clinical laboratory. And these facts are from Germany, so there are numbers from clinical laboratories, but also this applies to whole Europe. So for example, Institute for Laboratory Medicine in Rüsselsheim, Germany, this is in the Western Germany, is able to analyze 1.8 million laboratory analyses per year. This means 4,600 analyses per day, which are more than 1,800 patient samples per day. In northern Germany, we have a laboratory that is able to analyze 12,000 samples per day, which results in 150,000 single results per day. And in southern Germany, we have the Tübingen University Hospital, which is able to analyze 5,000 samples per day. And how can they do that? They use total laboratory automation, short TLA. In this picture, you can see how a common laboratory nowadays looks like. So you see it's totally full with automation, and this is a linked automation line where the samples can be proceed automatically. And you see here some few manual workstations, but all in all, most of the laboratory space is with these TLA. And a question that often arises is, what happens with the employees? Do they all lose their jobs? Of course, it's not impossible that they lose their job, but let me bring you to another point of view, because if we think about another industry, for example, the automotive industry, and if you think about the cars of the 50s, cars were very simple. So there was no big equipment, they had no safety belts, they had no airbags, it was just a thing with 4 wheels, if you want, and it was obvious that people manufacture them. Nowadays, if you think about our cars, that are more super machines, so they have so many equipment, many electronics, safety requirements that need to be fulfilled, and yeah, they are nowadays partly manufactured by automation system, and still many people work for these manufacturers. So what I want to say is that today TLA is on the one hand overcome the shortage of skilled personnel, but on the other hand, it allows us to do more complex, more diverse analytics in a less effort of time, and at the same time for an affordable price. And what do the employees? They operate the devices, of course, so the TLA, in case of errors of the systems or feeding the sample tubes. They have more time for more complex analysis outside the routine. They do it fully manual, as you've seen in the picture before, on the slide before. Or they utilize these partial automation systems. These are systems that are not linked to the TLA, but of course needs to be operated. And they have also more time for scientific innovation and can offer new support for physicians. So the benefits for industry, we have the efficiency and reproducibility, because every sample is traceable at any time, and we can do processes every time in the same way. Safety aspects, we can keep away our employees from toxic substances or from infectious samples of the patients. We have a win-win situation, because repetitive tasks are very good candidates for automation scenarios. And on the other hand, we are happy when we can automate tedious tasks in the lab. We can overcome the shortage of skilled personnel. I mentioned this before, and we have quality improvements. And I want to show you what is mean with quality improvement.
Sorry, Nicole. Who is the boss? Is it there? Is it Nicole?
Should I click admin?
Well, she's admitting it now.
Yeah, if you admit, please, and you can also say okay up. Yeah.
Okay.
And also the okay up then. Yeah.
Okay, yeah.
Yeah.
Thank you.
Yeah.
Perfect.
And maybe even you can transfer the host to.
Yeah,
You have to do it, and that you have to go to that participants.
Mm-hmm.
Yeah. You just show the participants. Click on it.
Mm-hmm.
And you can take if it comes down. Yeah.
Make host.
Yeah, exactly.
Mm-hmm.
Make host.
Okay.
Yeah. Now the mini will take care of it.
Perfect. Thank you.
Yeah.
So we were at the point of the quality improvements, and quality can be expressed by bioanalytical parameters in bioanalytics, and there are several. Maybe you know some of them. Here are a table. You can get an overview. What parameters can be improved due to automation, and what can't be improved but will be unchanged if you just translate your manual process into an automated process. So parameters like the accuracy, specificity, selectivity, or linearity are related to the method itself, and others can be improved due to automation. And all in all, we can say that lab automation can improve quality in terms of precise measurement data and extended the measurement range. So these bioanalytical parameters are validation parameters, as they are used for method validation in bioanalytics, and they are defined according to the ICH regulatories, validation of analytical procedures, text and methodology. So the most famous of them are for sure accuracy and precision. And the accuracy is the deviation from our measured values on average to the true value. And precision or standard deviation is the deviation of measured values from each other. And maybe some of you know these dartboards, where the black spot is our true value and the red little dots are our measured values. And in these two cases, you see we have good accuracy, because the red dots are on average close to our true value. And in these cases, we have good precision because our red dots do not deviate that much from each other. So in other words, we can say that the accuracy are systematic errors, and they are related to our method. And the precision is the random error, and that can be improved due to automation. So next parameter I want to show you is the limit of detection and limit of quantification, short LOD and LOQ. And it is defined as the lowest measured value at which an analyte can be detected or quantified. And this is a value that you can calculate as the 3.3-fold or 10-fold standard deviation of the blank plus the average of the blank. And here in this graph, you can see what that means. The green line is the LOD. So every measured value that is higher than the LOD can be detected, and every measured value that is higher than the light blue line can be quantified. So when we automate, what happens? We learned from the slide before that we can lower our standard deviation. That means when we calculate the LOD and LOQ, we come to a lowering process. So we can improve LOD and LOQ due to improvement of the standard deviation. So what does that mean? Our next parameter is the working range, which is the area between the LOQ and the saturation. And as we can lower our LOQ and LOD, we can also expand our working range. That means we can detect and quantify more or less concentrations of our analyte than without automation. Robustness is a parameter that I put in brackets because we cannot improve the robustness, so the sensitivity to interfering parameters, but we can put our system or our process into better conditions. For example, we can better control our temperature conditions. So working range can be improved due to improved LOD and LOQ. Okay, so laboratory automation is indispensable in industry, but can we also benefit from it? Of course, we can also improve our reproducibility, also efficiency, not in case of throughput, but in case of time efficiency. So we can do other things when our automated process is working. Safety, of course, we want also to work in a safe environment. Also for us a win-win situation because also we don't like tedious work. Quality improvement, also regarding the parameters I mentioned in the last slides. And sometimes we can reach a faster translation, meaning if I have an automated process in lab, the upscaling process to manufacturing is easier. So yeah, but there are also problems because if there were no problems, every one of us would have automated processes in their lab. So of course, first point is the costs. Not only the investment costs, also, for example, consumables or maintenance costs. So these are also all follow-up costs that need to be remembered. Flexibility. So TLA is totally not suitable for us. We want to be flexible. We want to do process A in the morning. Our colleague does process B after lunch, and we want to do process C in the afternoon. So we need flexibility in research. It's also linked to the developmental work because we cannot just translate our manual process one-to-one into automated process. Laboratory space, which is expensive. So if you think about an automation system that is under our laminar flow bench, so that does not work because we need this laboratory space for other processes. It's also complicated. So there are researchers or scientists that are afraid of using automated systems. If you think about liquid handling stations, you need some little programming skills, and yeah, that's not obvious or clear that scientists can deal with these softwares, for example. We have also sometimes technology gaps, meaning we are searching or looking for an automated system, but there is no system between the manual process and the fully automated process. It's an inhibition of innovation, so could be, meaning if I have a manual process, I can faster and more flexible change it than an automated system. And you can have headwind from other researchers. So not everyone will be happy if you come up with an automated system. All in all, we need a flexible, small portable, multifunctional device that is also cost effective and where we can use our conventional labware. So in other words, we need this. But let me leave you not alone with that. There are, of course, solutions for you. And let me make you an example with pipetting, because I think nearly everyone in life science do pipetting sometimes. And usually we deal with these 96 well plates, and we have to pipette different liquids. And therefore we use these single channel pipette, and if we are lucky, we have a multi-channel pipette. So if this would be the end of the story, every one of us would be happy. But in real life, it looks like this, and maybe this. And we can improve our process if we use electronic pipettes, because we can increase our efficiency if we use multi-dispense mode, meaning we aspirate in higher volume and dispense our wells stepwise. And we can improve our precision in the case of speed, for example, when we standardize our aspirating and dispensing speed. But of course, the confusion of wells and source vessel is left, and we have still random errors, for example, if you think about the angle of pipetting, which also plays a role for the precision. So there are also small systems that are maybe suitable, maybe not. Here you see, you can say it's a 96 channel pipette, where you can pipette a whole 96 well plate in one single step. And this is also the limitation, because you cannot pipette in every well and other liquid. Here is the price between 12 and 16,000 euros, and you can get a discount for academia, which is good. But of course, the limitations, we are not that flexible, and they use also own labware, not regarding the plates, but tips and the reservoirs. Then we have here the OT2 OpenTrans robot, which is a low-cost liquid handling station. Low cost because it starts at $11,000, but the price can quickly rises, because if you choose, for example, an 8-channel pipette, you are at $14,550, and if you choose some more reservoirs or tube racks, that are all additional costs that came up. And a very new guy on the market is GoodBot. It's equipped with a single channel pipette, and the pre-offer special price from last year was 9,500 euros, with additional costs for the software. I think that are additionally 150 euros per month, so it's not that cheap. They say they could improve regarding speed, but they developed it especially for research. It's small and portable, and it was not clear to me if there is a discount for academia. Also, the new price of this year that was not placed on their homepage. So, and what researchers do, and this is what I want to show you, they do DIY lab automation, because you see the systems that are offered are not very suitable for every case you need, and that's why they do their own laboratory automation. And here, for example, you can see Findus, which is an open source 3D printable liquid handling workstation for laboratory automation in life science. So it's a do-it-yourself liquid handling station, and they could reach a relative pipetting error of less than 0.3%. And they also show the realization of special processes in the field of chemistry and biology. And all in all, they had material costs for less than $400. Here is an example where the authors used the OT1, so the first version of the OT2 robot I showed you 2 slides before, and they could realize the accurately aspiring and dispensing of nanoliter volumes for less than $6,000 in material costs. In this publication, the authors published their FIL. FIL is pipetting helper imaging lid, which is an inexpensive, small, open source personal liquid handling robot that can be also used for microscopy in the area, especially for in cell biology. But researchers also do other things, not only pipetting. For example, I want to introduce you Meow, a microbalance autosampler, which is developed for automated weighing of powder. It looks like this. It's a shortcut from a video, so if you are interested, you can have a look. And here is a software-based automation system where the author linked two systems that are not from the same vendor. So they use an autosampler, interact and colorimeter from two different vendors. Also, we in our lab do laboratory automation in the low-cost field. And here in establishment of low-cost laboratory automation processes using Autoid and 4-axis robot, we created a system where you can applicate in different processes. So we show here 3 different applications. First, we show also pipetting, where we use this electronic pipette, and we showed multi and single dispensing. We also did an autosampler for AAS measurement. And we show inoculation of bacteria suspensions, where we inoculated bacteria from one source plate into two new media plates, and this was under sterile conditions. And I have a video for you with these 3 applications. And in this video, I want to show you why you cannot exactly translate your manual process into an automated process. You have to think outside the box. So for the pipetting process, we were not able to do the tip change. That's why we have to rinse our tips. Also for the autosampler process, the personnel usually wipe the steel needle after it was in a sample. And here we had to implement this washing station because we were not able to automate this wiping process. For the bacteria inoculation, we used barcode recognition, where the robot can identify the source plate and the 2 new plates. And instead of using the single-use inoculation loop, as it is in the manual process, we had to use this steel needle and use heat sterilization. Of course, with cool down time. And here you can see how the first plate is inoculated, and after the first plate is inoculated, the second plate, and in the end, the lids are closed. So now you saw that we changed our manual process, but our data could not, or we could not see a significant disadvantage due to our automation. So that was the same data. Yeah, now you might think that DIY is nice and cool, but not every one of us is an engineer. And yes, you're right. And I also mentioned before that lab automation can be very complicated. We need sometimes programming skills, and that is why there are also assistants in the lab. On the one hand, commercial available systems, and on the other side, also DIY. And we also did, for example, a lab assistant with a voice lab assistant. So it's called Rainbow, and it can assist you and your colleagues in the lab. And here in this video, you can see my colleague Larissa, and she's talking to Rainbow. First, Rainbow assists her with a protocol. Then she set a timer, and in the end, and this is what I want you to focus on, she talks to a device where she has no knowledge about the software. I hope the tone is on. No, there's no tone. So she said narrate protocol. Read plate was so I hope you were able to hear something. Yeah, so in the end she talks to the device, and we offer this as open source. Everyone can download it and implement it in their lab. I know it's not that easy, but we have also an introduction, so it's an offer. And you can also add other devices. So this is also possible. So now at this stage of my presentation, your thoughts might be very diverse. So some of you might think about a specific process they want to automate. Some of you want to benefit from automation, but don't know what to automate. Some of you might not be sure about whether to automate or not. Some of you don't want to change their running systems and want to keep it like this, is happy with the manual running process. And others might think about the lunch today, which is also okay. But all in all, what I want to say is that it's not possible to give a universal answer for each situation, because laboratories and processes are very individual, and whether and why to automate is multifaceted. So there are specific cases where we have to look at if it's good or not a good idea to automate. But maybe we can have an example. Just remember this guy. Maybe we can help him with his situation. So, but for now, do you know these game books from the 80s? So these were books where you could create your own adventure. Because you can decide after each chapter which way to go and which chapter to read. And that's why you can create your own reading flow and create your own adventure with different outcomes. And this is what we've done for the question, can I benefit from laboratory automation? So we published a feature article where you can have an aid for successful introducing of laboratory information automation. So let's have a look at our guy John, and let's go with him on an adventure. Here you can see an overview of our chapters in our feature article. So it starts for everyone with the theory part, and then we can go down regarding our requirements or regarding our situation to different chapters and end up at 4 scenarios. First, we have to revive an unused system. We have to purchase a system, removal of an unusable system, or no automation is at the moment recommended, which is also a good idea, because better we do not automate, we do not the waste of money, then so better we do not the automation, then a bad outcome for us or not suitable system. So what want John? John wants to automate the ELISA process. So let's go with John on his adventure and start with the theory part. And in theory, we have to check if our reasons for automation are valid, because there are good reasons for automation. For example, the improvement of reproducibility, improvement of efficiency, or the elimination of monotonous tasks. These are good reasons, and then we can continue with the chapter goals. But there are also bad reasons for automation. For example, yeah, okay, it works for others, so it has to work also for me. I have funds left that needs to be spent, or I just want a new device because I want to have a new feeling in my lab. So these are bad reasons because they lead to waste of money, unused devices, and the most worst case is a negative attitude to automation that could mean you exclude automation forever in your organization, and that would be yeah, very sad. So John wants to automate the ELISA because it's very extremely frustrating, because it takes long time. He needs to be completely focused, and at the same time it's monotonous. He would rather focus on the data. Yeah.
Yeah. Sorry, I have a question, sir. Just wondering, you say modernization is not a good reason to use automatization.
Yes.
But it's not. Isn't that like, if we modernize, we always want to things make more automatic in a way? But why doesn't that work?
Okay, so modernization is not should be not the reason why you want to automate, because you just buy an automated system and you don't know if it fits your process. So maybe it gets clearer when we have a look at the further progress of these case scenario, but it should be not the first reason for automation.
Yeah, because at the moment we are in the process of modernizing our institute. We are building new blocks of the institute, and so the modernization is very top on the agenda at the moment. But so we have to be careful here.
Yeah, maybe you should be careful. Okay, in case of, for example, electronic pipettes, that's no problem, because you can do something wrong with electronic pipettes. But in case of other bigger systems, you have to rethink, why do I want the system? Is it just because I want something new, or is it really beneficial for my process? Okay. So I think these reasons are good reasons for John. So let's continue with the chapter goals. So for the goals, you have to really look if your process that you have in mind is the worst process in your lab. So the process with the biggest automation pressure. And therefore we offer here, or provide here, a checklist where you can name your processes and give them a score regarding their bad properties. So here, for example, we have properties like monotonous, error-prone, dangerous, and so on. And you can choose your own score. For example, a process that is not very monotonous, I give a 1. But a process that is very error-prone, it's a 10. And in the end, you build a score. For example, if you sum up the points, and then you can rank your processes, and the process with the rank number 1 is the worst process, or the process that really needs to be automated. And this is also what John did. And okay, here in the end, we see the ELISA has the highest score and rank 1. And yeah, John told us. So it was good to have this black on white. So then we can continue with the preparation of technical feasibility. What does that mean? So we have to be clear about our process, because if you think about our process as one, we are very limited in searching systems that could be suitable for us. Here also we offer template where you can really analyze your process. And also John did. He said, okay, my ELISA consists of pipetting the plate, shake the plate, wash the plate, and these 3 steps repeating until I measure the plate in the end. So being sure about these steps, it's much better than think about the ELISA as one whole process. Okay, John is not really sure why he should do that, but I'm sure he will be happy in the end. So now he came to decision tree 1. In decision tree 1, we are asked if there is a commercial system available for our process. And that triggers us to do research. And John also did research, and he knows exactly what he wants. He wants, in this case, an liquid handling station from Hamilton. So there is a commercial system available, and we can continue with decision tree 2. And here the first question is if there is if the throughput is suitable for the system. He said no, it's not suitable, but maybe other colleagues can use it. Then we came to the question, can you use the system for other methods? Yes, of course. On the one hand, he can use it for other methods. On the other hand, his colleagues. Does running the system fits your budget? This also means not only the investment costs, but also follow-up costs, because if you have this complex machine in your lab, you have to maintain it. John says, okay, it's expensive, but it might be possible. So we came to the question, is the training effort feasible? And John did some research and find out that training for this system is multiple daily training, which is cost intensive. So he concludes it is not feasible, and it would exceed the scope of the application. And then we came to the question if he already has the system. So he has not. And then we can go back to decision tree 1 to do further research. So a few days later, John searched a lot and checked again his process from the table before, and maybe a washer will help him. So there is a commercial available system, and we can go again through decision tree 2. So in this case, his planned throughput is suitable for this case, and running the system also fits his budget. Training is feasible because it consists of the operating manual, and he don't have the system, so it's recommended to purchase the system. So, and here again is the process. With this automation system, with the washer, we can automate step 3, 6, 9, and 12, which is not what he thought about. So he wanted this fully automation, but better a well-considered automation than a fully automation that will not lead to our automation goals. So the follow-up steps are get familiar with the tool. So checking the operation manual and get the introduction by the representative. Can he optimize his process? So maybe he needs less washing buffer if I have the automated system. Be informed about the function of my device. This is maybe obvious, maybe not, because often our devices can do more things, but we forgot it, or because we don't want to use it at the moment, but in the future maybe our colleague wants to automate a specific process and want to buy a new device, but our device in the lab can already do it. Trained personnel is essential for the automation success. So we have to train an appropriate number of users. And we should plan for unscheduled and scheduled downtime, for example, in maintenance or errors. And of course, we should help others by using them, and for example, when we make visualizing notes in the lab. So these points seems to be obvious, but the experience shows they are not. So this is why I mentioned here. So, and this is what is my possible answer for everyone today for you. This guided aid for possibly successful automation in your lab. I mentioned before there are 4 different outcomes, so we can revive an unused system in our lab or remove it, which is also a good message we can purchase the system and have a founded basement on this decision, or no automation is recommended at this moment. But there are still DIY or tailor-made automation possibilities left in this case. And you learned a lot about the background, but there are many, many more information in our feature article, so feel free to have a look on it. We have also a reading flow for a so-called silent reader. So a person who has not a defined situation, but is just interested in this topic. So you're very welcome to have a look on it. So the take-home message for you, from me. We learned that laboratory automation is indispensable in diagnostics, because we cannot proceed the quantities of the samples, and we have analytical requirements on our diagnostic. We also want to benefit from the advantages, but we have totally other requirements. Devices are expensive and unflexible, and unsuitable automation can have negative consequences in our lab. So automation should be carefully considered, that we can have an automation success. And now I'm at the end of my presentation. I want to thank you for your attention. Here are the contact dates from me and my professor, which is the head of our research group, laboratory automation and bioanalytics. So maybe we can keep in touch. And now I'm happy for your questions and for a nice discussion. Thank you.
So, like, let me ask. Yeah. The voice recorder thing. Meaning that if we have a freshman to the lab, is it that friendly, or you need
of course
any specialized knowledge?
So
say like you have a very first PhD student to the lab, and you ask the person to run the experiment.
Yeah, of course. Yeah, of course it can help. So you saw it, Rainbow is able to narrate you a protocol stepwise. So it says one sentence, then it stops. And you can do the process. And after you finish the process, you can say, okay, go on. And Rainbow narrates you the next part of a process. And running, for example, this photospectrometer you saw in the end is totally without knowledge about the software or the device. That's possible. Okay. Yeah.
And you would have one headset for everything that you want to do. For example, you do the setup, you put one headset on the head of the feet. You don't need to even know anything. You just and then you take the headset out, and then you go to the real-time PCR. You put the other headset, and
For example.
Then you have, like this, you have made us to become robots. But ultimately, for example, in looking at your example of the ELISA, I think it is the wish of all of us that rather we go to the lab and we just have this sample, we want to find it out for antibody X, Y, Z. I think we can have it there, and we can just put it there, and it does everything, and then at the end of the day, we get the data out and say, okay, it's good.
Yeah.
Do you see it is going to that side? Is it becoming like that somehow? Even, for each of these processes that we do, and it's a lot of time consuming, and this and that, rather than we would have a box and you just put the sample and then it does it till the end.
Yeah. So for research, it is, as I said before, it's linked to many developmental work. So you cannot just say, okay, this is my process. I program it on a machine and the machine does it for me. I have to really look at the data. How is the automated process going? What is the automated data looks like? So at the moment in our lab, we have these comparison experiments on this liquid handling station and in the manual way, and we see there are differences. So we have a higher average of blank in the automated pipetting station, and so we have to rethink our process. So this leads not to a lower LOD and LOQ, but a higher LOD or LOQ, not because of precision, but because of the average of the blank. So it's not easy to automate process, for example.
Yeah, but the Ford company that you were showing at first, the line started, and it was not perfect. Now, 50 years or maybe 100 years later, now they have even replaced the human and the whole thing from the beginning to the end. But I think in the lab automation, we are also in the same situation.
Yeah, you're right. Yeah. So years before we did, for example, PCR by hand, and now we have these thermocyclers, and this will be the same, of course. Yeah. You're right.
I'm not sure if everybody can hear me.
Yes. Yes.
So let me also say my greetings to all of you. I'm happy that Nicole and Michael were able to join in Tallinn today. And thank you, Nicole, for all your insights in what we are doing. Of course, to both of you, Dulmini, Reza, you're very welcome to come to our place as well if you would like to have a look and yeah, see what we are doing in the first place. Germany is certainly not around the corner, but yeah, you're of course welcome. And yeah, I also just wanted to say I'm excited to hear and maybe see what we can do together in the future and what the next days over there at your university may bring in terms of insights, in terms of possibilities for possible automation projects. So thank you again, and yeah, hopefully you have a good time together.
Thank you.
Thank you, Nico. And then we have our second speaker, Michael Rupp. He is the founder of H-Box Robotics, a venture dedicated to developing and distributing devices for vendor-independent lab automation. He holds a master's degree in physics. Michael has been a part of the pharmaceutical industry since 2006, specializing in high-throughput screening, compound management, and data analytics. He currently serves as the Chief Technology Officer at stemick-lab, a customer relation organization offering services for pharmacological and toxicological researchers. So he's going to talk about lab automation in early drug discovery. So over to you, Matthew.
Thank you. Thanks for the.
Make that smaller.
Yep. I just
the first. Yeah.
This one.
Yeah.
So,
yeah.
Okay. Good. Just wanted to see me in the picture. So thanks for the introduction. I'm giving a talk about lab automation, early drug discovery. I try to make it as broad as possible, hoping to find parts that everybody can have a connection to. Oops, what is it? Need the pointer. Okay. So I'll give you an overview about early drug discovery, what is it about, what we are doing, the role of automation in early drug discovery, some nuts and bolts, really kind of deep dive into technological parts of it, some insights and devices and controlling. I show you also the basics of automated liquid handling, or let's say as a kind of overview about it. And I give you an overview of some examples of automated systems that I did in my career. So a few words about me. So we already had an introduction just to show you where I'm coming from. So as said before, I studied physics at the University of Constance. Always had a faible for biology and IT, and at the end ended up in pharmaceutical industry because it combined everything I wanted to do. So I had robotics, electronics, physics, so it sounded quite well, and it was also quite interesting. So I started with let's say, data analysis for high throughput screening. In 2006, took over the high throughput screening group of Nicomed, went into the IT, preclinical IT, and then 2030 started together with a former colleague of me, the hit discovery Constance, where I headed the IT and the compound management, which we'll hear later about. In 2019, I founded 8bot Robotics, where I also started a collaboration together with Tole and Nicole and Albstadt-Sigmaringen. In 2022, our company was bought by Axam GmbH, and I took over the role of head of automation. And in 2024, just very fresh, I started together with a friend of mine, a company called stemick-lab. I will just lose a few words and then I dive into the topic. So 8bot. The idea about 8bot is doing vendor-independent lab automation. It started all with the idea that I wanted to automate a process, and I had everything by hand. I had the device, I had the driver, I had the knowledge, I had the API. I even found a cheap robot, but I did not find a cheap gripper, which I could use. So every robot I wanted to buy came with a gripper which was suitable for the lab. But there was no gripper for the lab at that point in time. So I said, okay, do my own gripper, and the other company evolved from this. In the meantime, I did some projects for, let's say, big and small pharmaceutical companies in the field of mobile robotics. Things like IoT controls for laminar flows or for freezers and fridges. So for mobile robots, they cannot open freezers and fridges. So what do? You put a door opener in front of it and control it over the network so that a mobile lab robot can do or can take out buffers or whatever. And it worked quite well. This was a work together with also Roche and Idorsia Pharma in Switzerland. And they did automized cell culture maintenance over the weekend. So there was a robot running around doing cell culture maintenance, taking out buffer, medium, whatever from the freezers and from the fridge. And as said, our part was to automate the devices around it. Stemick, as said, founded together with a friend of mine. We're doing assays for tox and pharmacology testing, also stem cell derived human neuronal cells, lumen cells mainly. Starting now with radiometric assays, so and also serve in the field of lab automation and biostatistics and data science. Okay, I guess most of you have seen this before. This is the screening cascade. So the rational approach to drug discovery is nowadays mainly based on, let's say, high throughput screening. So you start with a target hypothesis. So you want to cure cancer, you want to cure Alzheimer, whatever, and you have to have an idea normally what is the problem in the body, what is the problem in the cell. So and this is called a target. So you want to target the cause of the disease. What I'm talking about here is just like small molecules, and if you want to invent a new small molecule, which could be a pharmaceutical compound, you're talking about a chemical space of 10 to the power 20 to 10 to the power of 200. So it's more, and then every single atom in the universe on combination you could have in your chemical structure. So you have to narrow it down, and the whole process is about narrowing down the chemical space to the.
We didn't share the presentation.
Sure.
Yes.
Okay. You share?
Can you stop it and share it again?
Stop share.
Share this one here.
That's great. Yeah. Okay. Sorry for that. Okay, I will not go through to everything again. Just like a few pictures of the gripper we are producing. So the gripper is like available for different robots. It's a standard like adapter for the robot. So you can buy any robot and put a micro titer plate gripper in front of it. So as said before, this is a screening cascade, talking about small molecules. You start with, let's say, a plethora of compounds which could possibly cure your disease, and you have to narrow it down. So the idea is to narrow it down to a drug that is approved by authorities, and it's normally just one drug. So we try to narrow down the chemical space from this high number to just number one. So how do you do this? The whole process takes about 15 years, 12 years, 20 years, depends on the candidate. You start with a hypothesis. You start to think about what could be the source of the problem. So, and then you start with a screening cascade. What you do, you have a library of compounds of different chemical entities. You buy them, you produce them by yourself. It's about, let's say, from 20,000, 3,000 to 4 million. I think Roche has the biggest one at the moment, about 4 million. And you try to make the experiment with every single molecule that you think could be available for a new drug. So you start here with about 1 million compounds and try to test in your single assay, does this have an effect on your assay? The compounds that you then select are called hits. So you try to select a hit where you say, okay, it has a positive effect on my biological assay. Could be a side effect, could be a false positive, could be whatever, but at least it has an effect. So normally you select about, let's say, 500 to 10,000 hits out of a screening cascade, and you have a closer look at them, which means you just retest them. You can retest them in single-cates or you retest them in multiple-cates. If you then find out that in principle you find interesting structures, which normally means not just the molecule itself, but also, let's say, the basic structure of the molecule. So how is it constructed? You're searching not just for one molecule that could be a potential candidate, but you're searching for basic structures in a molecule where you can tune it, because all the hits you find here are just like nice ideas, but that's all. Even patenting is difficult. All you get out from this one are starting points for chemists. So they say, okay, the structure is interesting, let's try to optimize it. This is called a lead structure. The lead structure, you try to find out, is it an interesting structure behind, the concept behind this is okay, and you try to optimize it. You try to optimize the solubility, you try to optimize the toxicology, and you try to optimize the side effect. And if you think you're good, then in the best case, you have a few candidates and you go to the preclinical phase with about 20. And then you heard it all before, I guess, in the clinical phases, you start with a few candidates and narrow it down from phase 1, 2, 3 until approval. The problem is it's extremely complicated and it's extremely costly. Who from you knows Moore's law? Ever heard of this? Moore's law, you know. So Moore's law, coming from the chip industry. George Moore was asked by a newspaper, so, what do you think? How will the chip industry develop in the 1950s, 60s, I guess? And I said, well, I think it will double the size or the number of transistors on a chip every 2 years. And the funny thing, it did. And the question here is it a self-fulfilling prophecy, or is it something like, well, it's just an intrinsic natural law or something like this? But it's really impressive. You see a logarithmic scale, and it just see how the integration of transistors just like boosted up on all this here, which is also often used as a comparison. So what would a car look like if the performance of the car industry would be like this? So it's really impressive, and every industry wants to do the same. You know this one? It's called Arum's law. So if you spell Moore's law backwards, you come to Arum's law. And this is the productivity of every $1 billion you put into a new medical structure, into a new pharmaceutical compound to get it to the market. So in the 50s, it's about $100 million you have to spend to get a drug on the market. And it doubled every 9 years. So it's exactly the opposite of Moore's law. So you get more expensive, gets more difficult. About the reasons, you can speculate. There are many reasons. Some reasons are for sure. You have the low-hanging fruit in the 50s. You have the rational approach to pharmacological research. You have a lot of regulations like here, thalidomide. So this was really a game changer. So this was also an introduction of, let's say, GLP, GXP guidelines in the pharmacological research. You have also some ups, like the first wave of biotech, some HIV drugs, and also, let's say, the orphan drugs event, where a lot of, let's say, neglected diseases where the big pharma was not interested before because the market was too low, were produced. Yep.
There is an issue here. The $1 billion in the 50s is not the same value as $1 billion.
It's already corrected. So at least I claim it. So I didn't make the graphic, but yes. But the trend is clear. So it gets more difficult, and it's just like not that easy. So And everybody is searching for a blockbuster, which will just like boost this company. So what can we do, or what was done also the last years? So increase throughput and efficiency when you really do something like screening. So data accuracy and reproducibility is also important, because if you deliver results which are not reproducible, you lose a lot of money. Integration of robotics and automated workflow was done in the, let's say, last 20 years. Resource utilization, also important. So you can buy a lot of devices, but in the best case, you should use them 24/7 and not just distribute them over different labs. Knowledge sharing and collaboration. For sure, it's not that complicated if you work in a small lab, but if you're working in an organization consisting about a few thousand researchers, it gets really difficult. So I'm talking to people, asking them things, and I say, well, perhaps we have such an institute here, we have just a device here on the campus, but I don't know, because it's just too huge. So you have to share the know-how you have on the campus. In the last years, we have the opportunity to go to high content and high complexity assays, not just a single readout, but let's say multiple readouts or imaging, or let's say something like cell cultures. And finally, it's all about reducing the costs per test point. So the idea is get bigger, make more experiments, and then it will work. If this is really the answer, I don't know. These are just like a few ideas. A small overview about early drug development, what is done in which phases. So you start, as said before, with a target discovery and validation. So this is brain work, I would say. So you go to literature, you go to biomarkers, you're really in the lab and you do basic research, mostly done in academia. And then you try to develop an assay, so a meaningful biological experiment that you can use to test the compound if it's working or not. If it's internal in a company, or if it's external, normally you transfer the assay to the automation group or to the guys who know how to work with it, and you try to replicate the assay in the lab. So it's just a one-to-one transfer to make sure that the guys who invented it and the guys who wanted to screen it or automate it can do exactly the same. This is very important, but it's seldom really working. This is really a big gap there to transfer from one lab to the other lab and experiment, which is also, you know, all the reproducibility crisis, where you say you have a lot of publication, you cannot reproduce it. Well, it's real life, so we experience it day by day. And assay adaptation, you try to adapt the assay to a kind of automated world. So if you have something like flicking plates in your assay protocol, it's quite difficult to teach a robot to flick plates. So you have to do something else. You have to wash them. You have to centrifuge them, whatever. This takes some time, and it's always a question about cost. So how much time do I invest to automate the assay, to miniaturize the assay? And what do I get from it? Typical example is a customer comes and says, well, I have now an assay in 96 well, can you do for 84 well? And we invest about 2 weeks for modifying it from 96 to 84 well. And then we say, okay, what about 1536 well? It's a fourfold higher integration density, fourfold lower consume of proteins, of reagents. And this is a question of time. It gets complicated. If you invest 3-4 weeks in a normal company, it's just like, let's say 50,000 euros you to spend, and then you have to calculate, is it really worth it to miniaturize it, or should I stay with the old format? Pilot screening is normally done with a small subset of the screening library. So what you do here is in principle proof that automation is working. So you start your automation and you say, okay, I take about 10 plates of my screening library and I just check out, are there results? Are the results looking good? Is the automation really working? You do it as in replicates, you do this in replicates on different days, and then you compare the data. And if this is stable enough, the customer normally says, okay, I go for the screening, which is then also more costly. Typically, you have a breakpoint here where the customer can say, okay, doesn't look good, I don't want to do it, so we stop now. Or we go to the primary screening, which means you screen your whole library or parts of the library in an automated way. Could be, as said before, about 20,000 compounds, could be 1, 2, 3 million compounds. Largest screen I did was about 1.5 million compounds in a radioactive method and a filtration-based assay. Hit confirmation, I also lost a few words before about this. You take the hits and you just try to replicate them. And in the best case, they show up again, also in different replicates on different days. They do not all the time. Typically, after this, you try to get a dose-response relationship. You try to reproduce the pharmacology or try to find out the pharmacology, and you're searching for compounds which have an IC50, in the best case, something in the nanomolar range. In the worst case, something a high micromolar range. But at least you get an idea, is it really something that is worth looking at? Because hit confirmation, you can also find some hits which are false positive. For instance, if you go for fluorescence assays, you have autofluorescence compounds and they show up at hits and you say, well, I got a new structure. And then you go for a dose response curve in a meaningful assay and you find out, well, it's just like an autofluorescence compound and it has no pharmacological effects at all. At the end, you have the hit to lead phase, and this can go from weeks to years. So chemists are trying to tweak the structure of the compound to add some parts of it, make it more soluble, make it more, let's say, more affine to proteins, whatever. And they cook, and every week, typically, you get new samples, and you report the results back to the chemist, and they try to optimize it. So. This is the same picture, just like with the automation grades. The blue parts are like manual work and the orange parts are fully automated. In early discovery, it's a lot of orange because it's just like a lot of compounds. You cannot do a lot of manual work. Target discovery, as said before, brain work, assay transfer. You have to take out, the manual parts of the manual assay. In assay adaptation, you do a few parts manual, but the rest is all like fully automized. Just at the end, hit to lead could be that you have also, let's say, measurements like you cannot do in a high throughput way, or you do not want to do in a high throughput way, such as permeability or solubility, whatever. Could be good to do it by hand. There are some parts you can still optimize, like say in the target discovery and validation. Big story at the moment. I know it's a buzzword, but it's AI-supported research. There are a lot of companies, they're doing research with artificial intelligence and trying to find out new ways for new medicines. And this is really a big story. And let's see where it ends, but at the moment it looks quite good. For assay transfer and adaptation, there's a cobot support, which is also, let's say, referring back to what Nicole said before. Having automation in the lab, which is usable without, let's say, a fully automated system, which I can use as a biologist, turn it on and use it like my mobile phone. What was the last time you read the instructions how to operate your mobile phone? 10 years, 20 years, 30 years. So I had a Nokia one time and I read the instruction. But today you never read the instruction. Why do you have to do it with lab devices? I know it's more complicated, but in principle, I should have a robot and I should tell the robot, go from A to B and pipette 20 microliters from A to B and then change the tips without having introduction and without having a training and without everything. So I think this is the best thing that could happen to the pharmaceutical industry to get the lab automation in this respect really to the bench. In the primary screening, you could also, let's say, enhance your system to increase the, let's say, the smartness of the approach. Typically in primary screening, you have your assay and you run the exact same assay over days and weeks because you want to have comparable results. What you can do is you select the compounds you want to screen depending on the results. Typical screening library is separated into, let's say, 3, 4, or 20 different parts with different, let's say, chemical structures like fragments, like protein inhibitors, like whatever. And if you see, well, I don't find anything in this library, I just switch to another library with different scaffolds. And at the end, last thing I could add to here is the AI-supported prediction of compound properties and also automated synthesis. I think this is already 10 years old, or let's say 50 years old, where the first fully automated closed loop research systems were installed. So you had a robot doing synthesis, You have a hypothesis how the new compound should look like. You test it with the same robot in the same cabinet on your assay, and then you go back in the loop and make a new hypothesis how the compound should look like. And 15 years ago, this was machine learning, I would say. If you now add on top of it artificial intelligence, this could really be a game changer. You see it also, we'll come back to it later, so, you know, the AlphaFold thing from Google. Huh? So it's not perfect. So I was talking to guys who are doing protein cryo-structured elucidation. And I said, well, it's good, but it does not make us work less, because it's a good idea for basic structure, what you're getting from AlphaFold, but it's not really the real protein. But let's see what's development from there. Just a historical overview about automation technologies. Let's say in the 1980s, I would say the first automated liquid handling systems at least have been in ideas in the labs. Not really viable, not really on the market, but let's say the first systems came in the 1980s. In the 1990s, there was the first rise of the high throughput screening and CombiChem. So the idea was at that point in time, we just screen everything we have, and we tell our chemists, cook everything we have. And the idea was combinational chemistry. We have our modules, our building blocks, we click them together and we find out the best molecule. Did not really work. So boosted up a lot of libraries. So there were libraries with a lot of compounds, but not really interesting compounds. LIMS system came also in the late 90s, so electronic handling of data. So the more data you have to handle it. Lab notebooks are nice and also widely used today everywhere you look. But if you have digital data, you have to collect it in another way in the LIMS system or ELN system, depending what you prefer. The microarray technologies, this was the time where I was stepping into the business, was a big thing in the 2000s. The idea is to put small droplets on, let's say, glass slides and do, let's say, DNA, mRNA experiments on there and do measurements there, and also synthesize in microarrays. Frankly, I don't know how much this is used nowadays, still present, but at least it's not in my regime. Next generation sequencing, where it was easily just to sequence your plasmid or your sequence, and also generate a new sequence, and then let's say not in months, but in, let's say, days or even minutes. Then the lab on a chip device, which is, I would say, still a good idea. There are some applications also in point of care diagnostics, but I don't see it a lot in the labs. So the idea is you have microfluidics, you have different, let's say, reservoirs, different reagents on your chip, and then you measure it directly on the chip. So and as I said, so I know it from the diagnostics, but not much from the early research. Machine learning, 2010s, perhaps also before 2010, started with image analysis. So the let's say easy part of it, so how to detect cells. So if I can teach a robot what is a cell, and if I can stain a cell, perhaps I can also look at the cells more in detail and get an idea about my drug, what my drug is doing with cells or with other systems. The 2020s, which was kind of unexpected for us all, well, COVID came, and also for lab automation, this was not a game changer, but it was interesting because a lot of people also from the university popped up and said, well, we need automation because we cannot also read these samples here and evaluate these samples. So I got calls from former professors of the university who said, well, can you help with us? So, and we provided them liquid automation just to get their assays done, because they were also doing the first measurements in the university, because nobody was prepared with this huge amount of swab sticks to be tested. And I think this gave rise to a little bit of acceptance also in the lab, so that to say that also in the labs, it was, let's say, recognized that automation is not just a bad thing and a strange thing, but it's really useful, which is also my experience. Normally you think about scientists, about people who who do just science, but in the lab itself, it's more handwork, I would say, because everybody has his idea how to pipette and how to treat a cell culture. And if I'm doing it, it's okay. If my colleague is doing it, it's wrong. So it's a lot of voodoo still there. And the people are saying, well, I have to do it by hand because I know how to do it. And well, this is difficult. Emerging technologies, few buzzwords here. What I see now is a lot of, let's say, also crystal automation, organ on a chip, which I find quite interesting. Also, 3D cell culture is quite interesting. But at the moment, I don't see a lot of, let's say, effect on the research quality. But nevertheless, there are interesting models for pharmacology. The idea would be you have a few organs on a chip and you simulate the whole physiological behavior of your system, which is working in principle, but did not produce meaningful results as far as I know for pharmacological research at the moment. And again, here AI. So I think it's a big thing at the moment. I had also a few customers who were just like AI-based companies who are buying companies to reproduce their results. So it's a big thing at the moment. Automation level, relaxing slide, just like what are the different automation levels. We have the manual automation, you know it all, manual pipetting. We have semi-manual, like Nicole pointed out, is exactly this Integra pipetting robot, which I find quite nice. This is, let's say, the easy, the low-cost variant where you're helping pipetting with 96 wells in one shot. There are also more complicated devices by Integra, which do semi-automation, where you can do a little bit of hit picking, dilution, and so on. And I did not use it, but I liked it quite much. I got a presentation, and I think it's a good device, but I don't know. Did you see this one, the Andrew Alliance robot? So they started with a robot grabbing pipettes, and over 3, 4, 5 iterations, they ended up with this robot here, now bought by Waters, with electronic pipettes, which has 2 arms and can do more or less everything that a scientist does. I know from a big company they bought about 50 of these devices, and I asked them, why do you need them? And they said, well, we don't think that it's used a lot in the lab, but we want to get the lab people used to automation, and perhaps this is a good start. And well, you have to be a big company to buy 50 of these. We have dedicated system. What I showed here is let's say dedicated liquid handling system, also from Hamilton, also a company I like quite much. They're good. They do produce good devices. They are focused in this part on liquid handling, and with this device you can do everything. So you can do automatic tip change, dilution, rack change, whatever you want. Like we also have companies, parts that specialize in compound management and also other medical devices. Then you have automated workstation. The line drawing here is a little bit difficult. So this one is a, let's say, pure liquid handling station. And this one is here a Cybio Felix from Analytic Jena, which is more or less a small screening station, something like this OT2 robot that Nicole showed before. And here you have different options. You have magnetic bead extraction, you have shakers, you have warm plates, whatever. So you cannot do everything with this device, but you can do a lot, starting from hit picking, automatic tip change, it's 96 well, 84 well, 1536 well plates, whatever. I would say about 100 to 200,000 euros, depending on the parts you try to integrate there. And at the end, you have this complex systems, and there you can put in everything you like. What you see here is an example from Hi-Res Bio. Quite nice concept. They have different modules in the hexagonal shape, and you move your modules to a central arm. So you need a device. You don't have to build a new system. You just slot it in to the robot arm and start with your new setup. Working well. Nice company. Very expensive.
Sorry.
Yep.
So is it like the same way that we can imagine here that the cost also, like as you move to the right side?
Absolutely, sure.
But does it go like linearly or logarithmically?
I would say at the last one it goes more or less exponential, I would say. No? So you start with the pipettes, pipetting it for 100, 200 euros, depending on the Integra, like Nicole said, 20,000, but I think it's a good price for it. The Waters about 60. The Hamiltons depends on, they have a small system, about 20,000 euros. Vantage, I guess so. Never tried it, but they also have a small entry-level market, but then it ends up at about, I don't know, 500, something like this. Analytic Jena, small dip, as I said, 100 to 200,000 euros, depending on what you build in. And at the right side, you have millions, so even if you start with a small system. Some technologies in automation. One part where I was working 10 years is compound management. It's something like the librarians of pharmacological research. You have to collect the compounds, you have to store them, you have to give them out, you have to prepare them, and you have to be very precise, because if you mix up compounds there, you end up in hell. So you could do research about 5 years and then find out it was just like this for starting compound. So, what you see here is a store that I operated, built by Remp. The guy formerly made robots for producing cheese. So he comes from Switzerland, and he was producing robots to move the cheese from left to right, which is quite difficult. And it was in the Swiss television. And next day, the guy from Rush saw it, and next day he called him and said, well, I need this device, but not for cheese. I need it for my compounds. And they teamed up together, and they built up this system here called REMP at this time. The idea is to store the compound at minus 20 degrees in a room with a robot, and the robot picks out a single compound at a single time and puts together your hits on one plate in a frozen state. The problem here is, or the discussion here is, in compound management are freeze-thaw cycles. Normally you have them dissolve the compounds in DMSO. DMSO takes up water. If you put the samples outside, they take up more water. So if you freeze them, the compounds precipitate in the aqueous phase, and the more times you do it, the worse gets your compound. So the idea is to produce the compound one time, dissolve it, and then just throw it one time and use it. And this was done with the system here. It's about 40 meters long, 5 meters high, and minus 20 degrees. So fantastically expensive. On the right-hand side, you see an incubator, an automated incubator. I think you also work with microtitter plates. And this is an incubator. You have this carousel inside. You slot in the microtitter plates, and inside there's a robot arm taking out the plates and handing them out over a small, let's say, door. Opening the door, handing the plate out, and closing the door again. So we can do also cell culture in there, or compounds, whatever you want. This is another device from Analytic Jena. This is a pipetter. So they're on the market since 20, 30 years. I would say it's something like a Volkswagen Beetle. It works all the time. So also not that cheap, but this is a multi-well pipetter, and you can add on different stacks to the left and the right. You can add on washing stations, pipette changers, whatever. So microtitter plate, just put it here to let's show it once again. You know, I guess so, the microtitter plates, format I work mostly are 384 well plates, 1536 well plates, as said before. Difficult sometimes, also like you produce bubbles inside, get out a bubble from a 1536 well plates, quite difficult. 384 well still works. Some ideas about how do we do quality control. Normally you have a positive control and a negative control on your plate. You have a normal distribution. You have the 3 sigma, where 90%, 99% of all your samples lie in, and you try to separate the negative and the positive controls as best as possible to get as less false positives as you can get. And you see here, this is a typical evaluation of data evaluation, where I try to define the cutoff and define the hits, where I try to select. Normally, all the plates are barcoded. You have a database where the compounds are listed, which compound is in which coordinate from which plate. At the end, you ask again on which coordinate from which plates are the hits, and then you select the hits, and then you get the compounds. Typical microplate readers, workstation based, I would say. This is a luminescence fluorescence reader. And the easiest way, you just slot in one plate manually, but you can also have the stacker solution here, where you put in, let's say, 10, 20 plates, and they're automatically measured. And there are different microplate readers, fluorescence, luminescence, radiometric measurements, whatever you like. So one of my first projects was confocal microscope on the high throughput screening line. Now it's, I think, owned by Perkin-Elmer Operetta. So you can also introduce let's say, a stained fluorescent cell culture as your model and screen on this in 1536 format. So whatever readout you like, you find a vendor who produces the device. Screening libraries, I mentioned it a few times before. Either you have your own library, you have your own chemists, and they cook for years or decades about thousands, millions of compounds, like the big pharma companies. They have their own libraries, which is more or less their treasure. There are a lot of companies who also provide you libraries, like Wuxi or Enamine, so they're just doing chemistry, and you can buy it from them. You can buy it from them in microliter format, or you can buy it from them in nanoliter format. And depending on what you like to do, you go to your trusted chemist, and he tells you which kind of structures you should use to find a potential candidate for a drug. High throughput screening platforms, the basis for, let's say, the work in pharmaceutical industry, are highly integrated platforms where you do what you like. So depending on the money you have in your pocket, you can buy or build your system who's automating also ELISA or other things. Typical high throughput screening platforms are built for 1, 2, or 3 purposes. So we have different devices which you can include in your process or not, but they are not that flexible. No? So you have to reprogram them, you have to evaluate and QC test the results. But they are not, let's say, a device which you can use for everything. Data analysis and visualization is also a big part. So I remember my first days in the pharmaceutical industry. I had a task to select some compounds having cytotoxic or genotoxic effects, and I started with a simple Excel list, about 20 compounds. I made it bigger, about 500 compounds, and then I tried to put it on a database and do the same selection on the database with a complicated, let's say, query. And for about 50 compounds, well, easy, was about 2 hours. But when I just used my whole library, about 500,000 compounds with a different, let's say, query, it would have used up 1.5 years just to do the selection. So handling big data sets is not that easy. So it's just like you don't think of it if you have, let's say, the data set of 1 million compounds, and you have to have an algorithm who's, let's say, not straightforward. So data analysis and visualization is important for automation. LIMS systems. LIMS systems and ELNs, so where to draw the line between both of them, I don't know, frankly. A LIMS system is good in your lab. You should know where your reagents are. You should know who produced them. You should know if they made problems, and you should know which data was produced. If you go into ELN, a lot of companies have ELNs, but it's still, I would not say it's not still well established in the pharmaceutical industry. A lot of people still have lab notebooks. The problem is also, let's say, with regulations. So how do I prove that my electronic system really is free from any manipulation, so that nobody can change data, etc.? And this is not widely established in the ELN community. And virtual screening platforms, like I said before, AlphaFold or docking mechanisms, been on the market since, I think, 30 years, where you have your protein and try to dock in different molecules, try to get affinity measures, and try to construct your molecule based on in silico methods. Going more a deep dive, these are typical robots which you find in the automation. Typical 6-arm, 6-degree of freedom robots, which you also see in automotive industry, for instance. Here one from Universal Robots, starting about 80,000 euros. Here one from ABB with my gripper on top on it, about 25, I guess, something like this. Thermo Fisher, SCARA robot. SCARA robot is something like a hand robot, just can move horizontally, which seems like a bad idea because I just limit myself to some movements, but most of the time in the lab, I want to just move horizontally. If I have the freedom to tilt my pipette, whatever, sometimes I don't like it. So most of the time I use a SCARA robot because I use microtutor, so it's good for that. It's not so complicated to program also. On the right-hand side, you see a SCARA robot from Bluebot, Chinese company. And on the bottom, a robot from Precise Flex, my most favorite one. It's also like a Volkswagen Beetle, just once. So they have a micro titter plate gripper and also different grippers. Special designs. This is something from Velocity 11, now Perkin-Elmer, I guess. So it's a completely different robot. I like it quite much because I did a completely de novo approach. I did a liquid handling platform. And what you see here is a robot running on a rail, going up and down, having a gripper, opening and closing, and also like with the smart mechanics, keeping the plate in parallel. So, but this is quite seldom that you see something like this. This one from ABB, two-arm robot. I talked to the guys and I said, why it's not widely used? And they say, well, the people are afraid of it. It just looks too human. But with two fingers, you can do a lot of things more than just with one. Here, the mobile robot. This is the mobile robot from Aztec, which I was a part in the project. This is a commercial available platform. And you see here also a universal robot arm. You have a gripper in front of it, and you have here a stereo camera. So with this camera, you navigate to Aruco markers. It's like 2D barcode markers, where you mark your reagents and your different labware. And this guy can run autonomously in the lab, take out microtutor plate, put it under the hood, delete it, control another device, take it back to the incubator, and do then other things. This one is Kevin from the EPA, also a commercial available platform, and a Precise Flex robot on top of it, and also doing lab work. So I never saw it in real use. I think it's still in development. Perhaps some is already sold at the moment. Small X-course to automated liquid handling. So you have contact-based liquid handling and contact-free liquid handling. Air cushion, typical pipette, you all know. Direct placement for, let's say, honey-like liquids, where we have to press them out directly, or direct displacement, where we have a working fluid, and the working fluid works on the air cushion. Contact-free, you have compressed air. There's a company called Pico, and they bought a device where you have small, let's say, small Eppendorfs with a hole at the bottom, and they shoot out nano droplets from this device. Micro syringe, also 20 years old, 30 years old, I would say. You have a syringe pump at the end, and you try to shoot out small droplets from an opening. Or inkjet, which is also used for small volumes. You can go into the picoliter range with inkjet technologies. Inkjet means you typically produce a bubble in water, and the bubble shoots out the droplet. Acoustic, which I will come back on the next slide, something I like quite much. So I was called 2006 from the vendor, and I said, well, I have a new dispenser, can dispense nanoliter droplets on plates. And I said, well, sounds nice, but I don't believe you. Come here and show me. So I told my chemist, compound management, give me the weirdest compounds you have, with the weirdest effects on liquids and everything. Let's see if you can do it. And it worked perfectly. We had this before, Analytic Jena, Analytic Selma, also a pipetta for 96, 24 well, semi-manual. Integra once again, and here's a combi drop. It's a peristaltic pipetta. I don't know if it's widely used also in your labs. So you have a cassette, and from this cassette you put in and can take out from a reservoir liquid, and you put it on microtutor plates. A microtutor plate is placed here. And with a cassette here, you just, with a peristaltic pump, dispense your liquid into this plate, and it's moved on to another columns when it's filled. This is an acoustic dispenser, what I talked before. At the moment, there's just one company who owns the technology because they bought all the companies linked to the technology. At the moment, it's owned by Beckman. This is an Echo, so the device is called Echo, and it costs about €500,000. The idea is you have a source plate here at the button where the liquid is inside you want to dispense, and you couple an acoustic transducer with water to the button, and it's an effect like you shake with a loudspeaker sand, something like this. You eject droplets, and you eject droplets of 2.5 nanoliter size. They fly through the air to another plate, to your destination plate, which is upside down on the other side, and they cling then to the plate. And it's working perfectly. It's just perfectly expensive. What you also can do is you measure the content of your plate. What you see here is a source plate where I marked the water content with different colors. So what they do, they exert a sound wave into the well, and from the echo that is coming back, they can measure the volume which is left in the well, and they can measure the amount of water and DMSO in the well from 80 to 100%. And it's about sub-nanoliter range which they can measure, and in a percent range, the water content that they can measure. Which is also good, because if you have a lot of water, you have a lot of precipitation, and in the worst case, your well is empty, and you should not even take this point into consideration. What you see on the right-hand side, I made a plate and put a heat seal on top of it, and I just spotted the droplets on top on the heat seal. The reason for doing this at that time was just like quality control to see if the system works, but it's also nice. You see the small droplets, like nebula droplets on the window, so which are shot on this plate. So normally you would take off the heat seal, and then the droplets would land directly into the plate. You can also do aqueous solution. It can do proteins, not whatever you like. You have to program it. It's complicated, but normally for DMSO and for water, you can fill up plates and then do nanoliter transfer, including hit picking, dose response curves, whatever you like. Slide about market and trends. I think you saw it also in the last 10 years. A lot of companies are bought by companies that are bought by other companies that are combined with other companies like Thermo Fisher or whatever. So it's a huge consolidation. Nevertheless, there are about 200 different vendors on the lab device market. So there are a lot of, let's say, independent companies who go into the market or enter the market. But as said before, a huge consolidation. For Industry 4.0 and IoT architectures, some examples. ROS, called Robot Operating System, is something like the Linux for robots. So it's an open system where you can combine different robot arms, devices, sensors, actors together, and build together an operating system for robots, which is not really prominent in the lab automation world, but which should be. You have the CELA 2 standard which I was working a lot of time with it. The idea is to have something like a common standard to integrate devices into your lab. If you have a headset on your computer, if you have a printer, you don't normally care which vendor it is, because nowadays you just plug it in and it works. You have USB and you have your drivers and it's okay. 20, 30 years ago, it was not that way. 20, 30 years ago, you had to install your driver, you had to find out which interrupt to use and whatever. And I see it at the moment, the same time in lab automation. You buy a device and you have to be an engineer to integrate it. CELA 2 has the idea to have on an abstraction level of IT, meaning over protocol, which you can run over Ethernet, over the internet, address every device, ask the device, what are you capable of? So ask a device, what are you? And the device says, well, I'm a shaker. What can you do? I can shake. Which limits can you shake? I can do 1,000 to 2,000, whatever. So the idea is that I have an open protocol where I have connected devices in the internet, in your local network, and they know each other. And I can integrate them as a file share on your Microsoft server or whatever. OPC UA, similar approach. Not so in the lab industry, more in the automotive industry. NEML, it's a market language for storing data in a way which I can find the data again, and I can also understand the data in a machine way. In an ideal world, this is the present, which we have now. Vendor login, a lot of, let's say, proprietary drivers and tools and machine controls, orchestration. I call everything like if I have different devices and I want to make them play together, like a shaker, pipette, or a robot, I would call orchestration. It's all the system of the vendors normally. And they're not open for, let's say, other providers or other protocols. In the ideal world, this would change and we would go to open standard. As said before, CELA, ROS, OPC UA, and for the orchestration levels, there are parts like Auto IT, like Nicole also used it, which you can more or less tune your computer and your lab device into a remote control device by just like replacing your keyboard and your mouse with a computer program. Sounds funny, but works. So the idea is that you install a program that can operate your computer and you orchestrate it from the background. Same for data. Data should be readable. Data should not be in proprietary format, and every data installed should be available, at least regarding the format. Last few slides, you can lean back. Just a few examples about fully automated systems I built in the last years. This is a system for producing acid-ready plates with this nanoliter transfer. Acid-ready plates we normally use in automation because you produce the plates beforehand, you hand it over to the scientist, and you just have a clear cut. So you don't have to have your compounds online in the system, but you produce the plates with the compounds that the scientist needs beforehand, hands it over, and then you're done. You can store it in the freezer, you can store it in the -2 degree, -20 degree, whatever you like or whatever you need. So the compound plate is ready with everything pre-filled that comes from compound management, which means your compounds or even your reagents. And this is a compound management station, which does nothing else. It has 2 incubators here, one for the source plate, one for the destination plate, and you can produce plates in microliter format. Total capacity would be about 1.5 million compounds in direct access, which I can have on my fingertips at my keyboard, and let me generate a plate out of this. Typical cycle times are about 4 minutes from begin to end. So if I want to have a plate, I tell the system, give me a plate, and it takes about 4 minutes until I have the plate in my hand and I can use it. This is the design for it. You see the robot arm in the middle with the whale. It's also a precise flex robot. It's an incubator, 2 automated incubators. It's an automated centrifuge. It's an automatic peeler, automatic sealer, a combi drop for adding reagents, and the nanoliter dispenser. So you see here the incubator, the centrifuge. It's important that you centrifuge the plates when you take them out. If you have bubbles, they're difficult to transfer or impossible to transfer, so you have to centrifuge them. And the next point, the seal, the heat seal is peeled off. It's just like a sticky tape you put on top on it, and then it's extracted from the plate. Shaker to get an even distribution of the compounds in the wells. A labeler, you label a plate automatically with a barcode. Acoustic dispenser, and here the peristaltic dispenser, and a sealer at the end. So at the end, we seal the plates, so we don't have any connection to the oxygen in the outside. And at the end, we have a scheduling software, orchestration software, which controls the different parts of it. This one is from Nicole company in Jena in Germany, and this system is completely based on CELA. So this is a system where you can take a device that speaks CELA and integrate it into here. For sure, you have to pay for the connector to say, so you cannot just use 10,000 devices. So this is the point where they're making money. They say, okay, we sell you a basic system, but for every separate device you want to add, you have to pay something. But the rest is open, so you can write your own driver, integrate it here. So you're not dependent on the vendor. You don't have to go to Thermo and ask him, please, can you do me a device driver for my washer, whatever. So what you see here is the software to program it. Typically, you have different labware, and you link the different labware to different stages in the process. This is real life. So this was just like 3D rendering. This is real life. Does not look the same, but it has the same functionality. So you see here the sealer, peeler, and in the middle you have the robot. All is housed in. Don't get dust out of it. This is another system. It's a screening system. It's a screening system with an incubator, with some liquid handlers, fluorescence, luminescence reader, and here flipper tetra from Molecular Devices. Quite nice device. What you can do with this is can do, for instance, luminescence or fluorescence measurements in situ. So you have a pipette integrated, you add on your reagents, and you can, in sub-second timestamps, you can see how your receptor opens up for calcium influx, for instance. So what you have here is, again, the robot arm from PreciseFlex. You have a cytomat who is handling out the plates automatically on this handling station, a barcode reader to read the barcode, a dispenser, combi drop, the plate hotel. Sometimes you need to incubate in room temperature, or you have to store plates, or you have, let's say, intermediate plates. Therefore, you use these hotels. A delitter, quite nice too. So to automatically take the lid off of a plate. Doing this with the robot arm is quite tedious. This one is a delitter works with suction cups and under pressure. This is the measurement device, the flipper, quite complicated device. You can have different plates inside, liquid handling, cell tanks even inside. And as I said before, you have a complete robot inside, and you can you have an amplifier for the rest light, and you can measure a complete 1536 well plate at the same point in time for every well in sub-second resolution. And here's a multi-label reader. It's in PerkinElmer Envision based on filters. System here is from BioZero, bought also by Bico, I guess a few weeks ago. And the reason I choose this, they also offer CLI connection. So I wanted to have a system where I'm not like locked in like from Telmo, where I just like rely on their drivers. Here I added also a few systems, a few devices working by CLI. So the system itself, it's like you can imagine like Windows or like macOS is working with their drivers. But now I came with a driver for this microdrop here. I wrote my own driver. I just could plug and play it in. So I have one time I can sleep well because the provider produces the whole system, integrates the devices, quality checks them. But if I have a special device to be integrated, I have an open standard, an open connector, and I connect it via CLI. This is how the scheduler looks like as before. You have different lanes here, I would say, for different labware. So this is one plate, and this is another plate, and this is the third plate here. And then you just tell the system, take them out of the cytomat, dispense here. You can configure the dispense, 1 microliter, 10 microliter, whatever, put them back for incubation, load them in the flipper, incubate them again, load them again in the flipper, put them back. And then you link these plates together. You tell them, okay, if the plate is here, you can put a mark here, start the measurement here, and put it back into the incubator. You can also have startup and shutdown processes like warming up of different components or whatever. What I see here is a simulation analysis. It's a Gantt chart. Quite important because doing one plate at one time with one setup, it's easy. So you can program it directly in every language you like. What's really complicated is having 1,000 plates started at the same and all the plates are treated the same. You can think about it like a train plan. So if you have 100 trains and want to have a perfect train plan so that every plane is at the right time at the right station, it's really complicated. And this is not easy. So and simulating this is a big part if you start an essay. Because if a plate gets stuck, you have to decide what should I do with the other plate. If it gets longer and longer, the plate gets incubated from 1 minute to at the end 2 days or something like this. And this is more or less the science and the thing that the companies are selling, a good scheduler. This is really important. You see here, this is also, let's say, the scheduler view where you can start processes on a 24/7 basis. Say in 1 week, I want to start this process. I tell the automation, I tell the biologist, put in the plates into Rec 1, and the system starts automatically, produce the data, collects the data, puts it on a data share, and then I go for analysis. All from me. Thank you for your attention.
So now the floor is open to the questions.
The systems that you showed is definitely good for a situation like COVID, and so that you have to do millions of patients to set them, or as you mentioned, for the when you have a very big compound library also. For us, I think we are more in the league of Nicole to somehow well, because we are, the whole reason for going to automation was for being able to somehow make some of the repetitive work that we do to become more affordable or to become more faster. But I imagine I don't know, probably the price of these systems should be millions or billions. I don't know how much it is.
So the systems here, so the systems for the as-ready plates, I did it in the own construction. If you go to the vendor, it will cost about 2-3 million, something like this minimum. So if you do it by your own and you know what you do, you get it for, let's say, 800,000. Yes, but it's still 800,000. It's not 8,000, absolutely.
They are more also for high quantities of the same thing.
Right.
Nothing wrong with it. But in a university setting, or as you mentioned, in a lab setting, so we need something that maybe we use it for one project for 3-4, I don't know, months or so, and then you can check it for the other one and everything. There's nothing wrong with it, but this is somewhat different thing for different places.
Which I also think is a sweet spot for lab automation. So, which is a thing that should come in the next years. It's like fusion. It's also coming since 30 years. But I think this is really something that would change a lot. I could take my devices, give it to somebody, not an engineer, not a techie guy in your lab, but just like my normal lab technician, and say, well, just program it. So, and absolutely, I agree to this. And I see there's a gap at the moment. I don't see a lot of devices that are capable of doing this. Even something like this OT2 robot, it's nice, it's good, but it's not that user-friendly as I would expect. Other devices are more user-friendly, like, let's say, the SciBio things, which are also good and nice, but they're then quite rigid. So the sweet spot lies between flexibility and rigidity. So, and also in between, you have to have the user acceptance and have to find somebody who's capable and wants to use it. So, and this is not solved at the moment. Absolutely. Yeah, I agree.
And then I have one question regarding acoustic distortion. I think there is some ways, but how accurately you pipe it? Like, I don't know, it was 2.5 nanometer. But what if I want more or less?
You should more. It's really quick. So for one well, about, let's say, 200 nanoliter takes about, let me think about it, like 3 seconds, something like this. So it's absolutely quick. You should just more. The limit is about 500 nanoliters. So then he has to remeasure. In principle, he can empty a well about 10 microliters immediately. Can empty a well about 2 microliters, but then you have to remeasure, and it takes long. But 2.5, the most time-consuming part of this acoustic dispensing is moving to the right well, because it's just like one actuator, measuring the volume and getting an idea how to treat it. If I shoot, then it's, I don't know, it's milliseconds, something like this. So it's really fast. So the more you like, the more you shoot. That's it.
Yeah, I think that how's the contamination possibility of the wells around the target one? Let's say
you mean shooting in the wrong well?
Yeah. Like
if the system is working well, none, absolutely none. So you can imagine integrated in the device, there's something like a static absorber. So if you have a plate and you have a dry air, you have electricity on the plates, and then the droplet is also, let's say, loaded with electricity, and then you get static deviation. But for this, you have an ionizer directly on it, and it's brushed off. But no, you don't have contamination problems. You shoot directly into the well. If the system is not working correctly because something is like broken, sure, you shoot in a lower well, but 1536 well plate, you shoot directly in the middle. Yeah.
Especially for us, I think the main thing that we want to do, especially with our, probably many other people also, is liquid handling, as you're saying. That's the most important. And the day it is always ending up with a 96 well plate or 384 plates, and so it is getting from a high volume to measured volumes into this, and then this reputations. Now, obviously, the step can do this, and we were very, especially the last 2-3 systems that you were showing, they are sophisticated and completely can do, and then do even more. But in our size, with your thing, do you think it is possible? Can we do, and can it be somehow, I mean, a cheap version, fast version, and that would be even, how can I say, with the budgets that we have, can we have something that would do it and would be better than your mini does it, or Dick does it, or I do it, or so? Do you think with your knowledge, what you see in the current market, is that a possibility or not?
So the question lies in between. It's a question of the budget. So if it's €1, no. If it's like 1 million, sure. And the truth lies in between. So if you're talking about a few €1,000, yes, it's possible. If you then say, well, can I do everything with this device? Can I do, let's say, different pipette tips, for instance? Can I have a 1,000 microliter pipette and a 2 microliter pipette? Can I exchange it in the right time? Can I have a multi-channel pipette and so on? So the truth lies in between. So it depends on your requirements. So do you have one process? Do you want to automate this process and want to press on a button and get this done? Sure. Want to be flexible? Well, then you have to decide. So should it be another robot? Should it be an adaptable gripper? Whatever. So
In the robot, and also I understand, if it is just one liquid dispensed in one, that is a matter of thousands. But if the tip wants to be changed, it jumps to 10,000, 20,000, 30,000. Am I correct?
You want a tip check?
Yeah.
No, not really. So no, not really. So it's more or less the flexibility. So if you tell me one process you want to automate it, there are ways to do it. So to say, there are cheap ways to do it, there are good ways to do it, there are brilliant ways to do it, and then you can choose. But if you say, well, I want now to use the system to do ELISA, I want now the system to do washing, I want now the system to do multi-channel pipettes, I want now to create 4-well plates, then you have to start up again. And this is the thing where you have to get smart and think about what are the things I want to do to be prepared. But doing something is sure possible. So I do not shy back for this. Absolutely not.
Yeah.
So what we done for Nicole, this was, what was this, 2 years ago? So
No.
I can't remember. So this is absolutely doable. So sure.
Because I was getting at the end a bit depressed. I was looking at it and thinking about my pocket. Yeah, it is not possible. But John, do you have any kind of application? You would have looked at it, you want to make it become automated.
My angle is immunology, but just the main problem is here, like the number of samples, right? So we don't have that many samples. We don't have like millions of samples, or what's like 12,000 samples per day, right? Not possible here. So, but at the same time, so yeah, if there would be a machine for 500 samples, that would be perfect.
What should this machine do? If I just may ask directly, because it's quite difficult to say what is it possible. So
it's a classical example. It's a ELISA.
Okay.
So ELISA, but it's again like 500 samples of ELISA. Again, not millions of samples of ELISA.
There are different ways, as Nicole pointed out, this OT2 robot, you start about 5,000. If you want to use it for ELISA, I would say it's about 12,000, something like this. And you can use it then, I would say. So frankly, I don't know the ELISA process in total, but I would say the most liquid handling process you can then cover with it. You have different pipettes there. You have, let's say, big pipettes there, and I think also multi-channel pipettes. But this is the range. Normally, you go to a vendor, so I want to automate the process. Then you're about 10 to 15,000 for this OT2 robot. How good it is, I cannot tell because I never used it. So, but it's an option. So, and it's also an option often used in academia. So a lot of people are buying it just like to try it out. So, if you want to go cheaper, you can go to cheaper robots like this Magician or this MG400, and you say you just want a pipette with a standard pipette, and you have the same process, or you have 3-4 processes. Then you're in a range of a few €1,000. This is doable if you want. So depends on the process. Sure.
And there's we're starting with a control program of one of the cattle diseases, that diarrhea virus here in Estonia. And then also also there will be like thousands and thousands of samples coming to the lab. So again, I can say, do we get the machine? Can we get the machine? But then we use this, can I benefit from laboratory automation? This algorithm and
yeah, maybe you should really think about automating just single steps. Yeah, and maybe we can keep in touch so we can discuss.
I think it's a good point. Taking the low-hanging fruits and not completely automating the process, but just like doing partly automation. This is also done in high throughput screening. So all the slides are shown here. They are true when we do it like this, but sometimes parts are also doing offline, like filtration of plates or something like this. You take it out because it's just too complicated. So there's a good point. So just taking the things which are really automatable and not having the idea automates everything. So. At you say thousands of samples. Question is, do they come every day? So do they come every second day? Do they come in one shot? Are they in vials? Do you have to unscrew them? Are they labeled correctly? So it depends on. So it's not easy to answer.
yeah.
But I'm free to discuss and delve into the process and the protocols to evaluate them.
This is exactly the way we are supporting that some of the parts of the process to make it to become less laborsome, less productive as this, and maybe that will increase our capacity to it. Taiwan has that. We have, for example, we have one compound, but we want to check it with several different bacteria. And at the moment, maybe we can do 2 per week. And so if we can somehow, with this supplement part of it, we can somehow make it more. But at the moment, we don't have also very long pocket, and we have to somehow take that power. But yeah, everything. 10 years ago, 20 years ago, I was dreaming that you can do something like this, but I never thought that it is within my reach. Now you are sitting here and say, yeah, you can. 2005, it is impossible to do that. That's so much, and who knows? I was in Poland. I was in Krakow, I think, in the airport, and there was one of the in the airport, one of the restaurants. There was a robot going around and was collecting all the dishes people were giving, and
it's widely used. Even in my hometown is about 400 people, and they have a small restaurant. And the reason they're using a robot because they don't have any personnel. So it's not about it's cool to have a robot because nobody wants to be a waiter and send the dishes out. Absolutely.
And I think if they can do it in the restaurant and so bring robots to do all the things, maybe we are slowly also reaching to all and even the mobile robots so that we can start to do.
In principle, so we had a research program with 8bot 3 years ago, and the idea was to have a mobile robot on the desk. And I was thinking, well, now we have self-driving Tesla cars, we have robots like the Atlas, or we have whatever, so should be easy. So let's buy a robot, go to the Git repo, let's install ROS, let's pay 2-3 master students, and let's have a lab robot. It's not that easy. The thing is, everybody, also Tesla and the other guys, they're inventing it on their own. There's no single, let's say, framework to control a mobile robot in a changing environment and having tasks. So, and all the automation here, I think there are about 4 or 5 companies now doing mobile robots in the lab. And still there it's difficult. If you go to an industry environment, you say you teach a robot, go from A to B, grab some things, even elevators are doable. That's quite fine. But the lab is complicated. You have a lot of glassware. You have to detect the glassware. The device is moved. You have stupid biologists in the lab moving devices. For a robot, this is catastrophic. Should be on the same place every time. So you have to recognize your device here. You have to have a 3D scan of your device. So is it really the device reader version 1 or version 2? Is the lid up or down or something like this? So it's really complicated. So sometimes I think self-driving cars outside are not so complicated than in the lab. But coming back to, let's say, simple lab automation, that's a question, where's the sweet spot? How big is the pocket, and how flexible you want to be? So, and sometimes there's a path where you can say, well, it's possible and it makes sense. Sometimes you have a system, but well, it's not really usable or it's not flexible enough, so it's not an easy to answer question. So that's also a reason we are coming here today to understand what are really the issues. So does it have to be sterile and so on. So this would be quite interesting to search. But having a robot here, like the one we did in Sigmaringen, so would be nice.
Yeah. And that is also, I think, even if you have the robot here, we are going to have reporters, journalists to come and have it in that we are getting to robotics and automation, modernization. It sells by itself if it goes on. And it's also good for, yeah, it would increase the speed. It would also, hopefully, it would be more accurate than what we are doing. Although listening to you, maybe you are thinking of getting things. But yeah, it is the stuff.
Happy to help. So I think we will go into labs afterwards and learn what you're doing.
Yeah. Thank you, Nicole.
Thank you very much.
Oh,
Please, no.
We have a certificate that you to show that you have been here.
Oh, wow.
We take also a lot of and we make also a picture of our